Okay, so let's talk about this gene regulation in prokaryotic cells. In prokaryotic cells, generally, we're talking about bacteria. Many times, genes that are related to the same function are grouped together on the chromosome, on a piece of DNA, in what's called an operon. So you did the POGO. So I'm hoping you heard some of these words, all of these words. So an operon is just a section of a gene containing whatever structural genes are relevant, and then the piece of DNA that regulates them. And the beauty of this is that this whole system can be regulated at the same time, so they act kind of as an on-off switch. So, and we'll get into the details of that for a couple different operons, the trip operon and the lac operon. And again, I think you should be somewhat familiar because I think you did the POGO. So, all right, so let's talk about operons. All of these are components of operons. And we'll go through each of these and talk about them, and then we'll talk about the trip and lac operon. Okay, so the promoter, just like in transcription, when we talked about transcription, that is the site where transcription begins. So that's where RNA polymerase is going to bind to start transcribing the DNA into RNA. And in this picture, it's shown sort of on the left, on the beginning of the operon. So lots of times we call that upstream. You know, the promoter sequence is upstream of the genes that are in the operon. Okay. In this particular example, the trip operon has 5 genes that encode 5 enzymes that all are involved in making tryptophan. So these genes are grouped together on this operon. And they're all important for tryptophan synthesis. Okay. And then the promoter is upstream. You don't have to memorize A, B, C, D, E. You don't have to memorize any of that. Just realize that they're grouped together for this common function. All right, and then this operon also has a place that's called regulatory genes. So in this red box, that sort of double vertical line cutting through the DNA, that just indicates that it's someplace else, like maybe not so close to the other genes in the operon. And in this case, that TRPR gene encodes a repressor. So we'll get to that. So this is a protein that's going to regulate the operon. And it's considered part of the operon because it impacts it, right? Acts as an on-off switch, contributes to the function of the operon. All right, and then the operator is a sequence within the promoter sequence that controls access of the polymerase and some other things to the operon. So this operator sequence is part of the promoter, and repressors and activators and inducers might all bind there, you know, to do different things that control the polymerase's access to the operon. All right, so that's the operator. Whoops, daisy, sorry. Where was I? The operator. Okay, and now we have something called a repressor. So this is a protein that's going to bind to the operator and stop transcription. So in this picture, this red like Lego piece is the active repressor. It's got a binding site in the operator. When it's bound to that operator, this white blobby looking RNA polymerase is physically blocked, and it can't get down that piece of DNA to transcribe. The blue box is the regulatory gene that encodes the repressor. So if this gets made, we get this mRNA, we get this repressor, which is the red Lego piece thing. And what's really cool and interesting and elegant is that tryptophan, the actual tryptophan, acts as a co-repressor, and it binds to the repressor. And when they're bound up together, that repressor gets activated and blocks transcription. So we'll get into more details on the trip operon, but keep this kind of in the back of your mind. All right, and then we can also have an inducer that facilitates transcription. And the way it does it is a little bit indirect. It takes the repressor out of play. So in this case, this green ball, this is a different operon. This is a lac operon, but this is just a general example of what an inducer can do. It turns on transcription of an operon by removing the repressor. So here's our inducer. It binds to the red Lego piece, which is the repressor, changes its shape, and now the repressor can no longer bind to the operator, and the operon, the RNA polymerase, can go. It can be transcribed. All right, so the inducer turns transcription on by removing the repressor. Okay, but then we have something called an activator, which can turn on transcription by directly binding to the DNA. So unlike the inducer that turns on transcription by removing the repressor, an activator can turn on transcription by binding to the DNA. So this is kind of like a transcription factor. We learned about those where it enhances the ability of the polymerase to get access to the DNA. This activator is similar to that. So it binds to the DNA and it helps RNA polymerase bind. Okay? All right, so we did the POGO. Let's talk about some specific operons that you did in the POGO, and we'll just go through some details. So this TRIP operon, okay, so the TRIP operon is what's called a repressible operon. The job of the TRIP operon is to produce the enzymes needed to make tryptophan. So basically the operon's job is to help tryptophan get made. So a repressible operon is one that's generally turned on, but it can be shut off. So if we have a bacterial cell that has a lot of tryptophan currently present, we don't want the operon turned on to make more tryptophan. Okay, that would be an energetic waste. The cell doesn't want to waste energy making something that it already has. Okay, but it can get turned off. Okay, so for example, I can never get my slides to move here. All right, when there's no tryptophan, the cell is going to need to make tryptophan. Okay, so the operon is going to need to be turned on. And so we want the repressor to be turned off. So what's happening here, here's our TRIP-R, makes the regulatory gene, which is the repressor. This repressor is inactive when there's no tryptophan, okay, and it won't bind. It's got a conformation that doesn't fit the operator. Okay, so when there's no tryptophan, this repressor doesn't work. Transcription can happen. We will make the genes that are going to synthesize tryptophan. So we want that to happen. If the cell is lacking tryptophan, it's going to need to make tryptophan. So we want transcription to happen at that operon. All right, but if we have tryptophan, we want to turn that operon off because, again, we don't want to make something that we already have. All right, so in this case, the regulatory gene will make this red Lego piece protein, which is the repressor. Tryptophan acts as the co-repressor, will bind to the repressor and activate it. And now the repressor has the conformational shape that will bind to the DNA and block transcription. So it's really so elegant and frankly beautiful, okay, that tryptophan is turning off the synthesis of tryptophan, right? So if we have a lot of tryptophan present, it's going to activate the repressor and turn off transcription of the TRIP operon. So that's what we mean by a repressible operon. Okay, it's normally on, but it can be turned off. So this is a negative feedback. All right, lac operon is another operon that we'll talk about. It's another kind of operon, and again, I think that was in the POGL. All right, this is called an inducible operon. And inducible operons are normally turned off, but they can be induced to be turned on. All right, and the job of the lac operon is to make the genes, or it has the genes, to produce the enzyme that break down lactose. Okay, so now instead of producing something, we're breaking something down. Okay, so it's normally going to be off. All right, so if we have high lactose and low lactose, right, let's think about this for a second. All right, if we have high lactose levels, we would like that operon to be turned on because we want that lactose to get broken down so the cell can get the energy out of the lactose, right? So under high lactose circumstances, the lactose is inducing the operon to be turned on for transcription to happen. If we don't have any lactose, under low lactose levels, we want that operon to be off, right? Because we don't want to make the enzymes to break down lactose if we don't have any lactose. That would be energetically wasteful. Okay, so here's a picture, all right? Here's the lac operon. Here is the regulatory gene. In this case, it's called lac-I. You don't need to memorize that. Here's its mRNA. It makes a repressor. All right, if there's no lactose present, this repressor is going to be active. It'll bind to the operator and block the RNA polymerase from transcribing the genes in the lac operon. So these downstream genes are not going to get made. Okay, so then lactose won't get broken down. So if lactose is absent, the repressor is active and transcription is blocked. All right, if lactose is present, all right, we want these genes to make the proteins to break down lactose, because that's the job of the lac operon, is to make the proteins to break down lactose. So if there's lactose in the cell, we would like the repressor to be inactive. Okay, so the regulatory gene makes the repressor. In this case, allolactose, I think it's an isomer of lactose, acts as the co-repressor or the inducer, binds to the repressor becomes inactive, can no longer bind to the operator, and polymerase can happen. So in other words, if we have lactose, or in this case allolactose, that acts as a co-repressor. It binds to the repressor and inactivates it, right? Then transcription will happen. The genes in the lac operon will make the proteins to break down lactose, which is the goal. That's what we want to happen. All right, so here's a summary table, okay? And anabolic and catabolic, those are words we've seen before. Anabolic means to build something up. So this is the trip operon. The job of the trip operon is to make tryptophan. Okay, so look over this slide, okay? If we have a lot of tryptophan, the repressor will block transcription because we don't want to make more tryptophan. If we don't have a lot of tryptophan, the repressor will be inactive because we do want to make tryptophan. Okay, catabolic means you're breaking something down. So the lac operon, the job of the lac operon is to break down lactose. Okay, so if you have a catabolic pathway, in this case we'll say the lac operon, high lactose is going to turn off the repressor because we want transcription to happen. We want those lactose degrading enzymes to get made. Okay, and with low lactose, the repressor will be turned on because we don't want to make those. Okay, so make sure you think this through and just kind of understand the difference between an inducible repressor and, an inducible operon and a repressible operon, okay? All right, and then there's of course another wrinkle in the lac operon system is what about if there's glucose? Okay, because glucose, if the cells have glucose, that's a lot easier for them to use. All right, glucose is a monosaccharide and lactose is a disaccharide. So if the bacteria have glucose, they're going to prefer to use glucose. It's going to be easier to do. All right, so hold on, I'm coughing. Okay, so we'll talk about glucose, whether it's what happens if it's present or absent. And then there's one more little wrinkle that is more involved in the rate of transcription, but not whether it's on or off, but the other system controls for sort of like volume control. All right, so here we go with the glucose situation, okay? If the bacteria cells do not have any glucose, they're going to need to use lactose as an energy source, right? So if glucose is absent, all right, the activator for the lac operon is going to be activated, and it's going to increase this rate of transcription. So remember, an activator binds directly to the promoter and facilitates RNA polymerase to transcribe whatever those genes are. So in this case, if there's no glucose, we want to make the genes or the enzymes that'll break down lactose, okay? So that's if there's no glucose and the cell is going to need to use lactose. All right, if there is glucose, that's a much easier energy source for these cells to use, like I said, and so we want to turn off the genes that are going to synthesize the proteins, the genes that make the proteins to break down lactose, because we don't need them, right? If we have glucose, the cells are going to break down glucose, okay? So if there's glucose, the activator is inactivated, which will decrease this rate of transcription off of this lac operon, okay? So I think just think that through and just make sure it makes sense in your head. Again, you're never going to have to reproduce any of these pictures, okay? But you'll have to be able to look at a diagram and figure out what's going on. Okay, so I believe that's the end. All right, awesome.